Hello, everyone. This is Dr. Juan from Shanghai Changzheng Hospital, China. I'm very honored here to share some data. Phase 1 open-label single-arm study of BCMA and CD19 dual-targeting FasTCAR-T cells GC012F as a first-line therapy for transplant-eligible newly diagnosed high-risk multiple myeloma. We know there has been a lot of advances in the last few years over multiple myeloma drugs, and people are living longer. But for high-risk myeloma patients, has a poor outcome with the standard first-line therapy, with the median overall survival is only about 3 years. So we really need a product we can give to high-risk patients to achieve the deep, sustained response and a more effective therapeutic option earlier in their disease course. Actually, CAR-T initially was thought this is going to cure the patients. Maybe the cure is not the right word yet, but the CAR-T has demonstrated manageable safety profile and high efficacy in the treatment relapsed and refractory multiple myeloma patients from other studies and our previous study with the GC012F. GC012F is a second-generation BCMA and CD19 dual-targeting CAR-T, which were designed to drive a fast, deep, and durable response in myeloma patients. This is a single-center open-label IIT study. Some of the key eligible criteria you can see are patients that have at least one of the high-risk factors. So to the right, you see the study design. All patients received 2 cycles of VRD induction. So after the patients were enrolled, they all get their cells apheresis before the patients are infused their cells. They got a lymphodepleting chemotherapy with the FC regimen. We treated 16 patients into 3 different dose levels from 1 times 10 to 5th power to 3 times 10 to 5th power per kilogram. The primary endpoint was safety, and the second endpoint including ORR, best response, MRD assessment, and the PK and the PD amongst others. This slide shows you the patient's characteristics. Notably, 94% of patients were evaluated as revised at the stage 2 or 3, 94 patients with a high-risk profile according to FSMART 3.0 criteria. And nearly 70% of patients with extramedullary plasmacytomas. So they are quite a high risk, and it might be poor outcome with the routine treatment regimens. So here we report the safety profile of this study. As you can see, the most common grade 3 or 4 event was the hematological, which is as expected, and the non-hematological adverse event was very uncommon. In terms of the cytokine release syndrome, all but 4 patients had CRS, which 25% of the patients having grade 1 or 2. No patients had a grade 3 or 4 CRS event. In terms of neutral toxicity, it was no any patients observed. The one thing I want to point out with this CAR-T is a very safety product. CRS could be resolved within 4 days and only in grade 1 or 2. We had the 60 evaluable patients, which we can see the MRD negative status, and the 40 patients were MRD negativity. And all the patients achieved the response rate. We can see here it's 80% of patients achieved CR and MRD negativity. Here we are going to discuss briefly the pharmacokinetics study. Every single patient expanded their T cells and they were consistent. So the peak happened about day 10, ranging between day 9 to 14. GC012F expanded well in all patients with a long persistent in all those levels, with a limited detection at 30 copy number per microgram DNA. The median peak copy number was nearly 65,000 copies per microgram genomic DNA. In conclusion, GC012F continued to show a favorable safety profile in newly diagnosed multiple myeloma, with 25% patients expand grade 1 or 2 CRS, and no grade 3 CRS and no ICANS or any neutral toxicity observed. Especially this drug project demonstrates an early and deep response observed in the high-risk newly diagnosed myeloma patients, which are 100% deep response. And all the patients we can see here, 100% achieve the VGPR or better. So this is a very safety project, and we can see 100 MRD negativity by Euroflow assessment. GC012F demonstrated the fast, deep, and durable response with a median DOR not reached. So we take together GC012F during targeting CAR-T cells shows very promising activity in transplant-eligible, high-risk, newly diagnosed multiple myeloma patients. Thank you.